This is Ash-Free Dry Mass and Chlorophyll-a, Analysis of Algal Samples lecture for ENR4345 Methods in Aquatic Ecology course at The Ohio State University. In this lecture, you will first learn the steps for analyzing ash-free dry mass samples, followed by learning the steps for measuring Chlorophyll-a samples. It is important that you become familiar with what each of these metrics measures, as well as the pros and cons of using each technique. When we analyze ash-free dry mass, we are going to be quantifying the mass of the organic material that we filtered onto the GFF filters during the last lab period. Ash-free dry mass is an inexpensive technique, and it is fairly easy to learn. However, some of the drawbacks of this technique include that it is unable to distinguish algal biomass from that of other organic materials, such as bacteria or fungi, and that you cannot tell if the biomass was viable or alive at the time of collection when using this technique. I'm going to quickly go over the steps for ash-free dry mass analysis. You can follow along with the Laboratory Analysis Week 2 section of your ash-free dry mass and Chlorophyll-a laboratory handout, which was the required reading for both last and this week's class. Remember that last week, after your ash-free dry mass samples were filtered, we placed them into the drying oven. These samples remain in the drying oven, which is set to 105 degrees Celsius for at least 24 hours. Next, the dried filter is weighed again. This will be the weight of both the dried sample and the weight of the filter, and this weight is recorded. Next, the dry sample will be placed into a muffle furnace, which will be set to 500 degrees Celsius for 1 hour. This will burn away all organic material, and what will be left on the filter will be ash and inorganic materials that don't burn, such as clay. Finally, the combusted sample will be weighed again once it has cooled, and this will be the weight of both the ash and the weight of the filter. Chlorophyll is a color pigment and is the primary molecule used as the photoreceptor in photosynthesis. This means it absorbs solar radiation, which is also known as light energy. Algae have other pigments that can be analyzed as well, including chlorophyll B or carotenoids, but that is outside of the scope of this class. Chlorophyll-a can be measured through spectrophotometry, which is the technique that we will be using in this class, or fluorimetry, which is an in situ technique that is commonly used. Some of the strengths of this technique include that it is relatively inexpensive and that you have the ability to tell algal cells apart from other organic materials, such as bacteria or fungi, which do not produce chlorophylls. However, this technique is more difficult and uses more resources than ash-free dry mass. And the amount of chlorophyll produced in a cell can change depending on environmental conditions. For example, in low sunlight conditions, the algae will work harder to absorb as much solar radiation as possible. In high sunlight conditions, the algae may not need to produce as much chlorophyll. There are many different techniques to extracting and analyzing periphyton and phytoplankton samples for chlorophyll-a. I will only be describing the technique that we are going to be using in this class. But if you go on to work in a lab, they may use a different technique. You'll remember that we put the GFF filters in the freezer after we finished filtering them in the last lab period. The freezing and thawing of these filters helps to burst open the cells, which releases the contents, including pigments, from inside the cells. Your instructors added 90% acetone to each of your chlorophyll-a filters, and then place them in a refrigerator in the dark for 24 hours. For this lab, we use acetone, which is also the active ingredient in nail polish remover. Think of this process as the acetone removing the color from the filter that we collected the algae on, so now it is a solution within the falcon tube. Just like how nail polish remover removes the color from nails. I am going to go over the steps for Chlorophyll-a analysis. You can follow along with the Laboratory Analysis Week 2 section of your Ash-Free Dry Mass and Chlorophyll-a Laboratory Handout. First, each student should practice pipetting. I myself like to do some practice pipetting before I start any analysis, because I find that it really helps improve my accuracy. Remember to wear gloves for this lab, both to protect our hands from the acetone and to protect our samples from being contaminated by anything that might be on our hands, such as soap or lotion. First, you will need to make a blank, which will contain only acetone. The blank will tell the machine what a 0 reading is. Next, you will pipette 3 ml of each sample into a vial, and that will be read on the spectrophotometer at both 750 and 664 nanometers. After those steps are completed, we will help you acidify each sample with hydrochloric acid, which would need to incubate for 90 seconds. The hydrochloric acid will degrade phenophytin, which is present in degraded cells but not in live cells. Finally, we will read the acidified samples at 750 and 665 nanometers. An accurate estimation of chlorophyll-a when incubated with acetone requires a measurement both before and after acidification to correct for phenophytin.